Hi everyone. So this is just going to be a video to finish off the sections of chapter 15 that we didn't have enough time to cover during our regular lecture today. So I backed up a little bit because we kind of stopped in the middle of section 15.6. So I'll go through 15.6, then we'll finish off 15.7, and then we'll be done with chapter 15. So this section is going to be on steroids. It's kind of funny to say like that, but this section is concerning steroids. Cholesterol, bile salts, and then steroid hormones. So I mentioned in the lecture, but we can make sure we're on the same page going through these, right? When we're talking about steroids, right, what I need you to recognize is basically, is the basic structure of what a steroid would look like. And this is usually called the steroid nucleus. So it's a series of cyclic rings that are attached together. And your body can decorate this nucleus, but don't think of it like the middle of an atom, but a nucleus just means like a center structure. So you have this steroid nucleus and your body has ways of decorating it with different functional groups, which will then change its function. So like I said, take home message from this is being able to recognize that four-ring structure. So we'll go through a couple of examples of particular steroids. The first being cholesterol, right? Usually we hear cholesterol talked about in a negative way. That's because high levels of cholesterol can lead to heart disease and then hardening of the arteries and everything else that goes along with high cholesterol levels. However, your body needs cholesterol to function, right? So either it's going to be used as a beginning point that your body can modify and then turn it into another type of steroid. And then cholesterol itself is also used in construction of cell walls and other signaling molecules. So your body needs cholesterol, it just doesn't need high levels of cholesterol. So you can either obtain cholesterol through diet, and it's also synthesized in the liver as well. So high cholesterol foods being meat, milk, and eggs, right? And then your liver is also synthesizing. So synthesizing meaning making, right, cholesterol. So a very commonly prescribed drug, I believe it's one of the most tolerated or one of the most prescribed drugs, is Lipitor, which is a small molecule that actually shuts down the synthesis of cholesterol in your liver. So theoretically, then the only cholesterol in your body would be that's what's already there, or any cholesterol that you intake in a source of food. When cholesterol gets very high, though, it tends to form plaques. So that's actually either the cholesterol itself is sticking to itself and forming large water-insoluble structures. Sometimes it can actually be embedded in the cell wall itself, rigidifying the cell wall, making it difficult for it to do its function. Another type of steroid that your body uses is bile salts. So again, we can see that I've just blanked on the word. Sorry, that steroid nucleus, right? We're looking at that four cyclic structure down here. It's been modified with certain functional groups that allow it to have its to function. A lot of these hydroxides here are to help keep it soluble in water, because this component is very highly insoluble in water. We also see attached to this group is more functional groups attached to the top, right? So we can see it's an amide, which is highly soluble in water, and we also see a carboxylate, which is again highly soluble in water. So the function of bile salt is that if you have a kind of a greasy molecule or you've eaten something that's not water soluble, what bile salts can do is that insoluble compound can bind, or not bind in the sense of making a covalent bond, but binds to it in a sense that it's attracted, right? So you have those intermolecular forces. Something that hates water will then be attracted to that hydrophobic point that steroid rings. And then what also happens is you've taken something that was originally insoluble, binded it up with that hydrophobic portion, and then the rest of the molecule acts as a way to take that molecule and bring it into solution, into water, because everything I have circled in red is highly soluble in water. So it's kind of a dual action type thing, right? It's taking insoluble fats and then bringing them into solution so your body can use them. So once fats have been, maybe if you've eaten fats, you need to get them throughout the body, right? Fat needs to be transferred from different areas of the body. Maybe either you're using it for energy or maybe it's being used to make cell wall, right? So it needs to get throughout the body. So how does that happen? Your body is 80% water, so there needs to be some sort of vehicle that can take these water insoluble compounds, move them around, get them to where they need to be, and then release them so they can do their jobs as fats, right? And so what this is done is through the use of lipoproteins, so literally fat proteins. And so what this will be is you'll make a covalent bond with some either fatty acid or a steroid, and usually it's complexed with a protein. And we'll talk about what proteins are. I know most people think of meat, but proteins are just a sequence of amino acids that make 3D structures. So they can attach these fatty molecules to themselves, and then basically it provides solubility. Those proteins are so soluble that even with some fat attached to them, they're still water soluble. And so that's how the fat gets moved around. So a common lipoprotein, you may have heard this before, right? We're talking about, so when they do a test for cholesterol in your blood, they're not necessarily looking for free cholesterol, though I do believe they can do that. A lot of the times you'll see low density lipoprotein, so LDL, and high density lipoprotein, HDL, right? So one of them is I forget off the top of my head, you can look that up, but one's the good cholesterol, quote unquote, one's the bad cholesterol, quote unquote. They both have functions in the body. It's just that when the ratios get out of whack, that's when you can start to get really bad heart disease, hardening of the arteries, and diseases that come from high cholesterol, right? So the way these lipoproteins work is that you have some sort of envelope on the outside that's usually water soluble. So what you're seeing here is a cross section of a lipoprotein. So you have these soluble outer layers. That means it can move through the water freely, and then packed on the inside will be those fats. So the fats like to stick together because they're hydrophobic. They like dissolves like, they hang out together. And then they're surrounded by this water soluble envelope. And that allows them to move throughout your body without precipitating out or coming out of solution. Other things that steroids can be used for, and I think this is getting more towards what most people think of when they hear the word steroids, which are anabolic steroids, right? These are steroids that are hormones. And so these are going to be the chemical messengers that can send signals throughout the body. So, right, your body doesn't communicate with letters and emails. It communicates through chemical signals. And one of them is hormones. So they're usually produced from cholesterol. And some of the most common hormones when we think of hormones right off the bat are sex hormones, right? Hormones is a very large category. Just the more common ones that most people somehow think of when they think of hormones are sex hormones, right? And that makes sense. They're produced in the pituitary gland in the back of your head or the front of your head, I forget where it is. And it sends signals throughout your body, right? And so the male sex hormones are testosterone and androsterone. And then you have the female sex hormones of estrogen and progesterone, right? We'll show on the slide in a second the difference in their chemical structure, right? And your body, again, I've mentioned this a lot, is really sensitive to the structure of these molecules. So even small differences have a pretty big effect, right? Other steroids you might have heard of are corticosteroids. So if you've ever had, sometimes there's infections. I remember one time I had, I forget what was in my lungs. I had something, and they gave me corticosteroids. They told me, don't worry, you're going to be studying all night. And I was up for, I think, 36 hours. So they're produced by the adrenal gland, and sometimes they keep you awake. Other times, depending on the structure, right, they're responsible for keeping electrolyte balance. So if your potassium and sodium and calcium levels are off, right, you can lead to disease states, and this helps keep things under control. And then also glucocorticosteroids. These are used to help regulate glucose levels. So not necessarily insulin, but things that would be part of that pathway of regulating glucose levels in the blood and inside cells. So what I have here, just some examples. So again, you can see how the structures of hormones are really similar, but these small little modifications your body can pick up on and actually have large effects on the body, right? So you're sending signals, chemical signals. Not that the only hormones are used for sending chemical signals, they're just a common group. And so again, what most people think of when they hear the word steroids are anabolic steroids. So usually these are derivatives of testosterone, which is the male sex hormone. And like what usually happens during puberty for men, increased muscle mass. It's basically hijacking your system. Giving yourself anabolic steroids makes it easier to put on muscle mass. But there are side effects, right? And you've probably heard of them before. We're not going to get into this, not like a sports medicine class or anything like that. But again, if you look at these structures, these are all anabolic steroids, really similar looking to testosterone. And again, they have the similar structure can bind to similar proteins in the body. So they're going to give off similar signals. So it's not all steroids can set off the same signals. They have to look close enough. So actually, if we go back and take a look at testosterone, oops, I went too far. Wow, that's going quick, right? When we're looking at testosterone, we see we have that carbonyl carbon down here and the hydroxide group at the top. And if you look at a lot of the anabolic steroids, well, there's my, oops, and it's having trouble today. Sorry, it's going a little bit slow, right? Again, we can see we have the same basic structure of steroids, but now I'm taking a look, there's my carbonyl oxygen at the same position. All of them have hydroxyl groups at the same position. Obviously, there's going to be some difference in chemical structure, but there is a similarity to testosterone, and that's why anabolic steroids have the effect that they do. So this is the last part of chapter 15 that we'll talk about. And so this is introducing the concept of cell membranes. And I believe bio class has probably covered this. This is probably covered in basic biology in high school, right? But when we're dealing with cell membranes, instead of having what we saw before is having a, when we had our fatty acids, right? One end of the fatty acid was a long carbon chain, and then we had some sort of acid group attached to it. A lot of the times there'll be a phosphate also attached to this group. That's why you may have heard of I can't think of the word, phospholipids, right? So it depends. There's a lot of different attachments that you can have, but effectively what a cell membrane is that one side of the fat is going to be water soluble, and then one side is going to be water insoluble. So we go all the way back, I think it's chapter 9 or 8 or something like that. Remember, like dissolves like. Instead of dissolves, I'm going to say attracts. There's no dissolution here. So like attracts like. So if we look at the structure of a cell membrane, that part that hates water will stick together, right? So actually what you're seeing on the top in the green is that polar head of the fat. And it's actually what you're setting up is if we have like a fat molecule like that, a fat molecule like that, it makes this bilayer, right? Bi being 2. So we have a layer on top. Let me use a different color. We have a layer on top, right? That is all, that's hydrophilic, meaning it likes water. So like the outside of your cells are bathed in water. The inside of your cells are also mostly water. So you have that water-soluble head of another molecule pointing down. And what they do is they make a section in between the inside and outside of the cell that is very fatty, right? And this is actually really important, and we'll talk about in a second. So if we can come to that fact that we've made this layer, so it basically is, it's like a skin. That's what the outside of the wall is. Not like a skin that you think of, but it's a protective barrier. It keeps the outside out and the inside in, because if it's attracted to the outside, it's probably going to be water soluble. Therefore, it's not going to want to get into that fatty layer in the middle. So then arises a problem. I have things that are outside the cell that need to get in. I also have inside the cell that needs to get out, right? So how does that happen? And we'll talk really briefly about different forms of transport inside cells. So this is basically the summary of what I was saying in terms of cell membranes, right? So they're used to separate cellular components. The picture that I had in the last slide is usually the cell wall of the outside of the entire cell. But if you are eukaryotic, which humans are, which means you have inside the cell, different functions take place in very distinct areas of the cell. Usually in like bacteria, there's still distinct areas, but everything kind of just floats around on the inside. Humans and other animals and eukaryotes have distinct, what they call organelles. And you've probably heard that before in biology. We're not going to get into that, but each individual organelle is also usually enclosed in a lipid bilayer. So it's almost like a Russian doll type thing, right? That was that summary we had there. So let's talk a little bit more about what's actually happening on that outside of the cell. And that's what I have down here. And it's not really to scale, but we have the green layer is the kind of like the outside of the cell. The blue is everything floating around on the inside of the cell, and that's called the cytoplasm. Again, that's biology. We're focusing more on the chemistry here. And when I was talking about separating organelles or separating compartments inside the cell, that's why you see like the nucleus here, and that's where the DNA would be held inside the cell. If you could blow up blow up means like zoom in on that outer layer, that green layer we see there, actually a lot of things are going on. And this is how cells can differentiate themselves from other cells, is kind of how they decorate their outside, right? Different fatty acids might be protruding out of the top. Cholesterol is also floating around inside that, that hydrophobic middle portion between the two layers, like here, right? That fatty layer in between. So a lot of things are going on. It's not just like a simple barrier, like a wall. It's very active, what's going on the outside of the cell. And so there's a lot of things going on. Just kind of pulling everything together, just like in one picture, right? We have our kind of our fat soluble things. I know it might be a new idea, but actually cells can communicate through the outside world, outside their cell. There's actually, they'll make carbohydrates. So sugar will be hanging off the outside of the cell, right? It doesn't always have to be sugar. Other molecules can hang out. And that's actually how the cells can talk to each other. So even though things are going on the inside, there's a lot going on the surface. So let's get to answering that question of, oops, why did it go backwards? Oh, never mind, I went forward. I was a little out of order. But the lipid bilayer, right? This is exactly what I was just saying on that last slide, right? That outside layer, there's a lot of stuff going on. There's proteins embedded in it, carbohydrates, and cholesterol also floats around. That helps give rigidity to the outside wall, because what I didn't actually define, sorry, is this idea of the fluid mosaic. Even though we think of the cell wall as being like stationary, it's actually flowing. It kind of moves together. That's why it's a mosaic. If you've ever seen it, it's kind of like a lot of things that come together to make one picture. It's basically what the cell membrane is. It is actually constantly moving around. So don't think of it like a rigid body. Some cell walls for plants are rigid, but like the cells in your body, for the most part, have this fluid mosaic where the outside actually, it's constantly moving. But it is still keeping things separated, right? And so, you know, how do we regulate how fluid it is? Because you really don't want it too fluid, right? That's why you can put things in like unsaturated fatty acid or cholesterol, right? So cholesterol will help, and proteins and carbohydrates help kind of rigidify the layer, but also having those unsaturated fatty acids help retain the fluidity of it. So it's kind of a balance between the two. So the last question we're going to answer is, okay, so I have this fluid mosaic on the outside. I have a cell wall. It's keeping the outside out, the inside in. But how do I get things in and out of the cell? Because the cells do need to talk to each other. They need to, you know, chemicals need to move out of the cell. Chemicals need to move into the cell, right? So how does that actually happen? And there's 3 different ways that stuff can get in and out of the cell. The first is diffusion or passive transport. That just usually means molecules that are soluble in water, but also kind of soluble in fat. A lot of drugs work this way. They'll just passively diffuse through the cell. Like there's nothing driving it. It just will, I mean, there's a low concentration. It's osmotic pressure, but it pushes itself into the cell with no energy involved. It's just saying, hey, I like hanging out in the water outside the cell, but if I touch the outside, there's parts of the molecule that are fatty enough that it kind of just sucks it into the cell wall, and then it'll just plop out on the inside of the cell. And then vice versa as well. It can just passively come through. Facilitated transport. What this is if you had your bilayer, I'll just draw it as 2 lines, and we had, say, a protein, and kind of, usually what they do is a lot of them will bunch up into like a barrel shape. So I'm kind of doing a cross section here, right? And what it will do is actually make a pore. And depending on the shape of the pore on the inside and the outside, and depending on how the inside of that barrel looks, it will basically gates what can come in and out. Sometimes whole proteins can go through these things, or you could passively diffuse, ions. What basically the idea is, it's facilitated because a protein has made a hole for whatever molecule you're looking at to pass through, but no energy is expended. And basically the shape of those proteins in that channel dictate what goes through. So it's not just like a hole and it leaks. It's a very, very defined hole. Only certain things can go through it. And then the last bit is going to be active transport. So active transport is very similar to facilitated transport. So again, we'll have our bilayer. There'll be some sort of, cross section of a bunch of proteins make a pore or a hole. The difference is, though, ATP is used to pump either out or in different ions or different molecules. So basically what it's saying is, it will usually, when things are passively diffusing, if we're looking up here for diffusion, right, it's osmotic pressure. It's just going to come through or leave depending on what the concentration gradient is. So if there's less stuff on the inside, well, it's going to move inside to try and increase the concentration. If there's less stuff on the outside, it tries to leave to increase the concentration outside the cell. So that's just osmosis, right? But what happens if there's a high concentration of whatever you care about outside and a low concentration in? Well, by the laws of thermodynamics, everything goes from high to low. So you're going to have to pay a price, and it's usually energetic, to take whatever molecule you care about and then force it to go against the osmotic pressure. And that's basically what it is it's waiting for ATP or some energy molecule, maybe GTP, whatever it is, to bind. That releases energy, and then the cell will allow something to pass through. So it's like an extra step in gating to make sure you're only letting things out and in that the cell really wants. And that's what I have here in terms of a figure. So we have our diffusion, where we have our red molecules, they just passively go through. If we have something like in the orange molecules, right, there's a protein channel, and that's what you see in bluish or purplish here, where it's made a hole, and a very selective hole, right? It's not like always allowing water and everything through. It's very selective, but those orange molecules can just come right through. It's facilitated because there's a particular gate or hole inside the membrane that allows it to come through. And then active transport requires an input of energy to start pushing things either out or into the cell, usually against a concentration gradient. So like I said, nature always flows from high to low. Well, you have to put energy in to go for something from low to high, and that's what active transport is. All right, so that's the end of chapter 15. We'll pick up with continuing our discussion with chapter 16, and that will be talking about proteins and enzymes.